Welcome everybody to our last Forever Green Fridays of the fall semester. We're excited to have Brian and Levi presenting today, as well as a lot of yummy food, which you guys should eat as much as you would like of. Thank you, Anne, for making the lovely salad. Thank you, Claire, for making the cheese coins and the cookies and the fudge. And also, Anne, like I say, one salad, it's like 8 different components. So thank you, Anne, for making many things.
Dietary consideration.
Super.
They're essentially the same salad.
Two different things. That's still a lot of work. Thank you. Let's see. We acknowledge our funders, which are on the screen in front of me. So it's MDA and Builders Initiative and McKnight Foundation and the Clean Water Amendment and that one and a lot of other wonderful folks.
CMR.
Thank you. That's CMR. Thanks to everybody joining online. We appreciate your attendance. Then should we have any announcements before we get started?
First, thank you to Claire and Anne for the food. And just a broader reflection on the semester. I have probably been more absent this semester than any time since I joined, thanks to baby Max. And it's probably been like the best semester that I've been around for. So I'm irrelevant and good job, Claire. And for co-organizing. Also, I want to highlight that many of you received an email on Wednesday asking you to complete a survey. So this is going to inform the Career Pathways program that we are developing. In a nutshell, this is going to be an effort to develop training opportunities, professional development opportunities, kind of 2 tracks, one for undergraduates, one for graduate students and postdocs, with a lot of focus on cross-disciplinary research methods, which is something we do a lot of, so it's a good fit. And hopefully it'll be a way to distinguish ourselves and draw in great students, and also to support those students as they progress in their career. So please do take the time to fill out the survey. And the last thing I'll say is that we thought we might get it out today. We didn't quite get there, but should be Monday or Tuesday at the latest where we can post our co-director position, and most importantly, call for nominations for the co-director position. So we're asking for nominations not really as a gatekeeping step, more as a recruiting step. We know that it's, taking on a role like co-director of Forever Green is not trivial. Everyone's very busy. So we want the community to help encourage the great leaders in the group to step up, put their name in the hat, or hat in the ring, whatever the right analogy is. And so that'll be coming out soon. And that position is about a 20% time, comes with a small salary augmentation. And it is for the folks who are eligible are current tenure track U of M faculty members. Because we're transitioning from a time when we have 2 co-founders who are senior tenured faculty members, and we want to maintain some of that stability in the context of our academic hierarchy that we exist in.
All right. Good afternoon, everybody. It's a pleasure to be here on last Forever Green Fridays because we always get that great food produced by some of the crops here. So I welcome you all. And for those of you who may not know me, I'm in the plant pathology department. I'm a Lieberman Okinawa chair. I do a lot with wild germplasm and helping to identify resistance genes. And I also want to introduce Nick Metz, who you probably know, and he's a research specialist on Kevin Smith's program with OAT, but coming forward into March, he'll be doing part-time looking at winter Durum breeding as well. So this is actually the first time that I presented to the Forever Green group. So I'm going to kind of take a step back and give you a little bit of a history about how this all started. But I just wanted to kick off the first slide by showing that our team has grown quite a bit over the last summer. So we started out in this program in 2013, and it was basically me and my technician just saying, like, let's plant out some Durums in the fall to see what might happen. But over the course of the summer, There were a lot of other people who joined on to the winter durum program, which I'm very pleased to see. So we have Nick coming on with the genetics and breeding in March. And then Jake had come on last year actually to trial some of the winter durums at various places around in the cropping trials. And then we were pleased to see that Fernanda and George, who have the interest in some of the nutritional and food quality, join onto the project. And then, of course, Kevin Smith was really the inspiration for this project through his winter barley breeding program. So he serves as a collaborator and a consultant. And then we really benefited from Colin, Catherine, and Matt's efforts in terms of trying to facilitate adoption and also scaling and potential outreach to producers. And then finally, one last thing I want to mention is Linda Dykes at USDA Fargo, who's been doing our durum and pasta quality evaluations. So just a little background about durum. It's Triticum turgidum subspecies durum officially, and it's a tetraploid species. So it's a hybrid from the A genome species of Triticum artu, along with the B genome related species of Agelos feltoides. Of course, all of you know its main product is pasta, so very popular in Italy. But if you go across south of the Mediterranean and North Africa, a lot of that will be used for couscous and bulgur. But also some durum flour is used in bread as well. So as far as durum wheat in the United States, it makes up just a very small portion of the total wheat, just under 5%. And most of that acreage is focused in the Durum Triangle, they call it. So that's North Dakota and Montana and the adjacent areas of Canada. So that's the heart of that Durum Triangle. And there is some what's called desert durum that's grown down in Arizona and California. So the production was about 80 million bushels back in 2003 over about 1.7 million acres. Average yields are about 37 bushels per acre. But what's really notable about durum is the price paid per bushel. You can see that in some cases, it's almost twice that for hard red spring wheat. And I've put comparisons with the other cereals there. So oats just at 374, and if you can get a contract with malting barley, you can get up to 660. So it's very attractive in terms of the price paid for producers for durum. So I was a faculty member at North Dakota State University during the start of the fusarium epidemics that area has seen, and it was very difficult being a plant pathologist without any immediate solutions for growers. But starting in 1993, the fusarium epidemics hit hard. And it affected all three of the main crops of hard red spring wheat, barley, and durum. And durum was really hit the hardest. It was really susceptible, so they lost a lot, and they continue to lose a lot. And unlike hard red spring wheat and barley, durum doesn't really have a lot of great resistance sources to move forward with resistance breeding to fusarium. But in addition to the biotic constraint that we had with fusarium, durum also suffered from an abiotic constraint, and that was a very severe drought in 2021, where there was almost a drop of 50% in yield. So a lot of these very serious production problems that we have in the spring durum area. So the question is there a possibility of growing winter durum perhaps in the north? This could be an alternative cash crop and also help diversify the agroecosystems. And as you know, with all the Forever Green Initiative attributes that provide those ecological services. And then the additional thing for farmers would be that higher price per bushel that's paid. But we need to have some of the durum production be spread out to avoid some of the risks. And that's something that would be really advantageous to have greater durum production in other areas. But the main question is, do we have sufficient winter hardiness in tetraploid wheats to develop a good winter durum variety? And that's really what the crux of my work on this project has been. But cereals certainly can be a very reliable winter crop for the bold north, as I call it, especially since winters are starting to get warmer. So it may be very feasible to have more of these winter cereals be viable crops. We all know that winter rye and winter bread wheat are very good crops, and they survive very, very well in most years. And we just need to have more of these crops on the acreage if possible. What about barley? Well, about 15 years ago, if you asked somebody that you were going to try to grow winter barley in Minnesota, they'd pretty much think you're crazy. because there'd never been any reports about people growing winter barley and having it survive reliably. But there's been major improvements in regards to winter hardiness in barley that have been made by the Smith Project. So Kevin has been doing this for about 13, 14 years now, and about 60% of his preliminary yield trial entries have greater than 80% survival. The other thing about winter cereals, and in particular winter barley, is that you can often see higher yields than the comparable spring types. In fact, Kevin explored this particular aspect by planting the same lines at the same site, both in spring and fall in Minnesota. And in some cases, the fall planted parts of this trial were yielding 50% greater than the spring components. The other thing that we see oftentimes with barley is winter barley will have greater malting quality than the spring types. And as a result of all this work that Kevin did, he released the variety of Equinox, which is a 2-row facultator that has good winter hardiness. So there's definitely a good example of a crop where there really wasn't thought to be any good winter hardiness that would survive Minnesota winters to a type where we really do have a viable crop. So the question is, what about durum? I guess I was, really inspired by Kevin's work to think about another crop that might work well in what I call the bold north. And so we began to think about winter durum. And so I had to look back in my emails, but I found the first missive that I sent off to Harold Bockelman, who is the curator at the National Small Grains Collection. I said, with the success of winter barley, is there anything in the collection called winter durum? And he looked and he said, yeah, there's about 98 accessions. And I just picked some diverse geographic regions of trying to get a few accessions to try. And he got them sent to us right away. So even though the email was on September 20th, I planted at the very end of September. And these were planted right here in St. Paul. And then we took stand counts from the fall just to see how the emergence would be. And then we pretty much forgot about it until spring and the snow melted. And we were quite surprised to see that about half of the lines survived the winter in Minnesota. So this was really the genesis of the project. This was what kickstarted the winter durum breeding project. And we did a number of tests over several years because we didn't just want to go on a one-off evaluation to start crossing. So we waited until we had about 3 years of data, and then the first crosses were made then in 2016. And at that time, Ahmed Salam, who many of you know, was on my project working on this particular program. So we wanted to expand beyond the USDA collection because it was relatively small in terms of winter durum. So we contacted some of our Italian colleagues that I've worked with on rust resistance. Many of you might know this name, Roberto Tuberosa. So he's a graduate of this department, was with Ron Phillips. With a name like that, he should be working on potato, but he's actually working on durum. And so Roberto was kind enough to send some of the germplasm that they had in Italy. And we evaluated that in Minnesota. And as you would expect, there wasn't a lot that survived, but that's not the point. The fact is that there are a small set that do survive really well in Minnesota, and that's all we need to move forward in our breeding program. And we also began to send out our feelers more to get additional germplasm when we got in contact with Friedrich Longen, who is a winter durum breeder in Germany. So he sent us 50 of his breeding lines, and they were really exceptional. I couldn't believe how good these survived, but we had an average survival of 95% in Rosemont and 97% in St. Paul. Generally, we get a little better survival in Rosemont because we plant in a shelter belt area that catches snow. So we usually get better survival there. But in this case, St. Paul edged it by a little bit. But we're really excited about these lines because of the exceptional winter hardiness, and also they have very good pasta quality. And then the final germplasm panel that we were working with was from Icarta, and that was the global durum panel. So they assembled this very diverse panel that included cultivars and land races, also the primitive emmers of the dicoccum, the wild emmers, dicocoides, and some other wild relatives. And we increased that in St. Paul to get enough seed. And then we did the first evaluation in 2022. And again, same sort of graph. Many of them didn't survive, 0 for many of them. But the fact is we had a number that did survive at 100%. including some of the wild emmers, which is quite surprising, being this is, largely Mediterranean species. So the end point is that we have a number of materials that we can use to breed for winter hardiness in the winter durum program. And we have started a little bit in terms of exploring the genetics of this winter hardiness trait by genome-wide association study. So the global durum panel has 90,000 SNP markers that we're using to map the loci conferring winter hardiness. So we're hoping that will help us in terms of breeding for winter hardiness in the durum. So I next want to hand off the next part over to Nick, and he's going to talk a little bit about parental selection, breeding, and quality testing. And then I'll finish up after that.
Well, perfect. So yeah, I'm kind of taking over the breeding kind of portion of the project here. And one of my first tasks has been to pick apart Ahmed's brain. So if you've ever had to pick up a breeding program from another breeder, sometimes it's like speaking a different language. So Ahmed had created some of these first initial crosses from the lines that had good winter hardiness. And he crossed it almost exclusively with Alcamo and Carpio, which were elite spring varieties from North Dakota State. And the idea there was to bring in the higher quality from the spring varieties and crossing them into the winter varieties. And I believe this was done in 2016. So many of these varieties have now been advanced throughout the years. So between 2018 and 2022, Ahmed collected winter hardiness data, agronomic traits, and disease tolerance. Those 30 winter hardy accessions were selected for population development, crossed with even more spring varieties, again, to try to bring in more quality tests or quality traits. So again, this is just kind of what I've been gleaning over Ahmed. He's advanced these populations and has found a range of winter survival throughout the years. The biggest thing that he's been really lucky with is those early winters were pretty mild, good snowfall, and that contributes to better winter survival. So his five advanced lines that he's been working with now are the WDA1 through WDA5. Those were planted in 2024, I believe, in St. Paul and Crookston, and had good winter survival. And pointing that Crookston is obviously much further north, so it surviving up there was pretty impressive and gave us some hope that maybe even further north it'll survive. There was some 100% survival at both testing sites, so those were advanced to 2025. And then 2025 happened. So these new F2, F3, and F4 populations were planted in St. Paul, and this is when I finally showed up, and I was here in 2025, and that's when we had very little snow and extreme cold, and this resulted in nearly a 100% loss. So you can see the plots. So the plots in the middle there is the wild emmer population on the left, and then to the left of that plot is winter barley, and to the right is winter durum. And then the plot picture taken in January 28th, that was all the winter barley that nearly had 100% kill. So typically, the end of January, you're hoping to not see your plants. You're hoping to see a lot of snow. That gave us an indication that this was going to be a tough year for winter survival. And it was. Most of the barley, I believe, was 100% winter kill. And with the durum, we had 78% of the plots were winter killed. However, there were a few survivors out there and stragglers, and this gave us insight to maybe these are the best of the best. This is what separated the boys from the men. These were the best varieties. So we had to think of something intuitive here because they didn't want to leave these plots out in the field. They were ugly. They were single plants. And so Kevin and Ed came up with this idea of shovel breeding, to go out there and rescue these survivors and dig them up and bring them into the greenhouse. When Julian was here, he had the idea of maybe attempting to cross these as they came into the greenhouse, but lining up the spring varieties just didn't quite work out. So many of these varieties or line survivors were just increased, and I'm hoping to plant them out actually this spring and see if we can get an increase and get them back out into the field for next fall. So we really want to, when these harsh winters happen, it isn't always the worst of the worst, right? We can still kind of have a positive spin on it and really maybe get some really hardy lines out of this. So after winter survival with winter durum, we really had to start looking at the quality. And so this was just my spark notes of quality testing in durum is when durum is milled, there's semolina, which is the coarse, kind of, it's not flour, it's semolina, it's coarse, that's what you make your pasta out of. But there's still flour being produced as well. And durum flour typically isn't used in a lot of products. We're mainly looking for the semolina. The other quality testing is for you want high test weight, high protein, and then this concept of falling number. Does anybody know what falling number is? I didn't know it. Well, Jim knows.
Yeah.
So I'll explain falling number the best I can. Falling number is done by this fun little machine down here that you create a slurry out of ground durum, and that there's a plunger in that machine, and it goes and it falls to the bottom, and you see how long the plunger takes to go to the bottom. And it measures the gluten content. So the longer it takes to get to the bottom, the higher the gluten is. And in pasta making, that's exactly what you want. So you do want a higher falling number. If you have a low falling number, there's typically alpha amylase production, and that happens in pre-sprouting. So that's typically when your quality goes down. Ash is when you have bran contamination. You want low ash numbers. And then another big one in pasta is your color. You want your beautiful yellow pasta. And so there are 3 different parameters within the color. Your L is your brightness, how bright. I'm going to pass this around. So as you're looking at some of these colors, this is winter durum. You want a nice bright kernel. The A indicator is your red-green indicator, and you want that to be low. You don't want really red durum. And then B is your yellow-blue indicator, and that's what high. So you again, this nice bright yellow durum. And the quality data was done by Linda Dykes up in Barba at the USDA. So in 2023, this is just some preliminary data that Ahmed got back. As you can see, we check out very well. And then Montana and North Dakota are the spring durum varieties. So that's kind of our benchmark where we want to be at. As you can see, the test weight, the falling number, and the protein is pretty good in 2023. But then some of the color and the ash tells me that our grain is a little darker and a little more, there's a little bit more bran on it than what spring varieties happens. So I was very curious about this data, that these falling numbers, because what we had actually heard, well, there's it. Who did that? Now it's mainly for aesthetics. That question is, it's primarily for aesthetics. They want their pasta to look as uniform as possible. And so when you grab a box of pasta, you want that nice bright yellow pasta, not a brown or red looking pasta. So the falling number in 2023 seemed difficult or different. I wasn't quite sure, because we had heard that winter durum actually had bad falling numbers. So in 2024, we sent it up there. We actually got these numbers where the falling number isn't quite up to the benchmark where spring durum would be. So seeing these numbers really gives us kind of a breeding target for falling number and for better color. But with test weight and protein, I'm pretty happy with that, because typically, those are benchmarks that we're looking for, especially in red meat. So with the quality data, Brian has reached out to a couple companies.
Yeah, so through Colin and Catherine, we began to make some contacts with folks locally here with the Perennial Pantry, and they're very excited and very interested in this. And also this past fall, we also had a nice meeting with folks at Barilla. They were based in Italy, and another buyer in Chicago. They also expressed a great deal of interest in having a local durum production. So one of the problems we have is even though the interest is high by these organizations, unfortunately, our seed stocks are not. They're low. So we can't like afford to give away seed for quality testing because then I don't have any to give to Jake for the cropping system trials. So that's been a bit of a problem. And I think back to the very funny story that when I was at North Dakota State University and Jim Anderson was the winter wheat breeder, Raleigh Sears gave him a very good piece of advice, and that is breed until you run out of seed. And now I understand exactly what Raleigh meant when we had this 2005 fiasco where we lost so much of our breeding lines. But I think it was a very unusual year, and I'm optimistic based on the 8 prior years that we're going to have a number of good years for producing seed. But how many people have driven down to Ames, Iowa, and have seen this great big huge Barilla plant along the freeway?
Yeah.
Yep. So where's the closest Durum field? 780 miles away in North Dakota. So what do you think they're paying for transportation costs? So if we can get folks at Barilla interested in producing a local crop of winter durum, that would be really great and economical for them. Relating to that point about the pasta color, I know probably all of you have eaten whole wheat durum before. It doesn't look like the regular durum does it, durum pasta. So it's dark. So maybe people like Perennial Pantry would be more likely to say, we're going to try some of this artisanal pasta made from winter durum, and we won't have to worry about the color so much, but it remains to be seen. So Catherine did a lot of sleuthing on the Barilla website, and she found that their sustainability goals are very much aligned with the Forever Green initiative. So we had a very productive discussion with the folks at Barilla, and we hope that we can connect better to demonstrate to them that we have something useful that could be grown in the Minnesota area in the future. And just for a final slide, we're also trying to do outreach to producers. So we had planned a field day last year, but because of the extreme die-out, we thought it wouldn't be advisable. But we are planning for the second week of June to have Winter Durum Field Day. So we'll be inviting all the Forever Green folks here and also extension educators, agronomists, producers, and users to this event. So we hope we can generate more excitement and interest in growing winter durum. So just to summarize, as I mentioned, some of the winter cereals have several key advantages over their comparable spring sown cereals, and it would fit really well in the cropping systems that are sustainable in the northern tier states. Of course, the cold weather in Minnesota makes it a challenge, but as I showed you in some of those germplasm panels, even though most of those panels are surviving at 0%, there's a small percentage that are very reliable in their winter hardiness, and those are the ones we're using in the breeding program. And we have made some advancements already in the breeding lines to have winter hardiness, good agronomic traits, but we're still working on those quality traits, as Nick alluded to. But I think with a few extra crosses and populations, we'll be able to maybe release a good advanced durum line that could be released to growers. So with that, I just want to acknowledge all the people who have kindly contributed the germplasm to this project, and then to the folks that have worked on this project in my lab, starting out with Ahmed and Julian Cooper for a very short time, and now Nick. So with that, I'd be happy to entertain any questions that you might have about this program. And thanks for your attention.
I have a question online. When is the typical harvest date for winter durum in Minnesota?
Yeah, so that would be about mid-July. So barley is a little better. So barley first week of July, and it allows for double cropping more easily. We're going to be about 10 days to 2 weeks later than barley. And I was just talking to Jake, and I was asking about possible double cropping, and there are maybe some options. He was indicating that maybe like hemp or other things could possibly go into that scheme, but it's still going to be a little tough with that later maturity.
What are your plans with the wild emmer?
Oh, so the wild emmer, that's something that I've worked quite a bit on, because in my position, I'm to work with wild cereal relatives. So we started a program in putting together a wild emmer panel, and we've evaluated that and identified new rust resistance genes to all three rust, stem, stripe, and leaf rust. So now we want to just see if there's anything that's useful in terms of winter hardiness. And preliminary indication is that there's a few accessions, surprisingly, even though it's a species from the Mediterranean basin that have winter hardiness. So we'll be trying to cross that one into some of the lines to see if it has different alleles for winter hardiness. So yeah, so we have that panel in our lab if anybody wants to do anything further work with the wild emmer.
How far out ahead of seed availability for producers do you want to get with the producer?
Oh, that is a tough question, Les. I mean, we got so burned in 2025, and then just trying to come up with enough seed for Jake to plant in a cropping trial. I go, please, if there's any left over, give it back to us. But I'm probably not the best person to ask that question. Maybe somebody who's agronomist can do the math for how many years it takes to blow up enough seed to give to somebody who's growing foundation seed or whatever. So I really don't have a good answer for you.
Question about the falling number. Since, to my mind, pasta and bread are like vastly different in terms of what's going on structurally in that product. So is falling number just as important for pasta making in Durham as it is for bread making with red wheat, or is it different?
Yeah, I mean, when we talked to Barilla, he talked about the elasticity of when they do mixograph stuff as well, which is another quality thing. So yeah, in Barilla, they really that was almost the most important part.
So I guess you want the pasta to not fall apart in your
Right.
pasta pot or on your plates.
Right.
Same concept, maybe.
Yeah, we don't have a word for it though.
Yeah, did we tape that?
We did.
Okay, I have to look back at that Zoom call. Yeah. But
Yeah, he had an Italian word.
So was it Barilla that said the falling number test is an indicator of gluten?
That's what we did. Yeah.
I've never heard that before.
Yeah.
Because in other wheat classes, it's used as a measure of alpha amylase activity.
Right.
So an indicator of whether it's sprouted or not. So if it's a more viscous solution, then it's sound, and that plunger takes longer to go through. But if it is higher gluten, I could imagine the plunger would take longer to fall
Yeah.
through also.
Yeah. No, it's just from me briefly skimming as well and looking at pasta quality in the United States.
one other thing I forgot to mention is that in Kevin Smith's barley program, he's been able to identify facultative barleys with a high level winter hardiness. Right now, it seems like most of the best ones look like true winters, but we are exploring other ones to see if we can identify those facultatives that have high level winter hardiness. Why is that important? That will help speed up the breeding process immensely because we're not having to vernalize, and we can get multiple generations than we could with a true winter type. So we're
Plus, you can spring plant your trials and
Yeah.
make lots of seed.
exactly. Yep. So
Pretty-
we're
pretty
looking
good.
for that facultative Durum that has the highest level of winter hardiness possible. I've been asking all the Durum breeders about that, and I'm not quite sure about for Minnesota conditions. So
Speaking of that, you mentioned, Nick, potentially taking some of the seed produced in the greenhouse and planting in the spring. I guess would that be early enough to get it vernalized? Or did I mishear you when you're in the fall?
No, and I don't think so. I don't think it'd be fast enough. That's our big thing, is my first thoughts are how to get 3 generations a year like we do in small grains.
Right.
And it's gonna be tough. Harvesting in July and getting it turned around to getting it planted back in is real quick turnaround.
we have looked, Julian did a little experiment on speed breeding to see if we could help hasten this process a little bit. So we'll try that again in the coming year as well.
Yeah, with cannellini and pennycress, those teams can sometimes just plant as soon as the ground thaws, and then you get another cold stretch, but they must have a much shorter fertilization. period.
Yes.
Yeah, I'd say minimum of 6 weeks.
In
Something.
barley, we always do 6 weeks.
Yeah.
Another question online, haven't you, if they're too high, a falling number?
That I don't know. I didn't get that when I was looking around online, so I'm not sure. Yeah.
I don't know, Jim, do you know?
Anything above 400 is considered sound
Right.
or kind of as good as you can get. And a lot of times they won't even report numbers beyond 400 or 500 because the plunger is just taking forever.
And keep it, when we say 400 and 500, that's seconds.
Yeah.
So,
And I think for spring week, you start getting docked if it's below 250 or 300.
okay.
So
Okay.
between, even above
Yeah.
300 is still, you don't get docked, but 400 is considered into the gold.
Yeah, the few companies I saw was 300 in Durum.
Have you spoken with Bakersfield Flour here in Minnesota? They're a very small local mill.
We haven't
They're in Northeast. Do you know where Diane's Place is, the restaurant?
Yeah,
The record store.
exactly. I've been over there. Thank you for that. We'll try to reach out to them. Yeah, thanks for that.
Got a question from Matt Lovett. As I understand, Durum is produced way out west to control fusarium. Is that a trait being looked at in the winter lines, or is winter less susceptible? Also, high contract prices.
That is a great question, Matt. Thank you for that. So one of the things that I thought about with winter durum is that it possibly would go through the heading phase at a time when there wasn't as much moisture like we see in like midsummer, so that maybe we could escape a little bit the most prone times for fusarium. But 2 years ago, I looked at a few samples. So the sample that's being sent around looks like really nice amber durum. But the one that I saw from 2022 looked a little gray, and we isolated fusarium from there. So we're definitely not going to completely escape fusarium. And there are some sources. I was talking with Stephen Shu out at Albany USDA that he has some sources of durum that look like they're fairly good for resistance to fusarium. But we also need to think about where we can grow this. So Jake was talking to me about establishing a trial in Roseau. And why Roseau and Durum? Well, up there's really no corn. So corn is a big harbor of Fusarium inoculum, and they also get a lot of winter cover. So we can get our snow cover. So we get good survival, and we're in an area where there isn't a lot of maize and other small grain cereals. So maybe we can do some things to help augment durum getting through any infection periods of fusarium. But it's going to be tough, especially since the tetraploids seem to be a little bit more prone to infection. So this is a good point, Matt, and one that we're really thinking about in the future.
Does that mean that there's resistance on the D genome?
I can't remember.
It's mostly on the A and B genome, but I think Durum is just so super susceptible that a lot of the resistance genes coming from common wheat they're up against such a susceptible background.
It just says it's not enough.
Yeah.
Yeah.
Okay, so I am Levi Teigen, come to you from other corner of campus, food science and nutrition, emphasis on the nutrition side. So this is a project that I did with George and then Abby Johnson over in public health. And our question that we were trying to answer essentially is what happens when you eat this stuff. We were looking at what we focused on was the gut microbiome essentially. So background for the project, at the point of this, there had been no attempts to study the effects of specifically intermediate wheatgrass on different measures of gut health. We know fiber is important to health of the microbiome, to our gut in general. We know that these intermediate wheatgrass products contain more fiber than traditional whole wheat products. And so the question was, does eating these compared to whole wheat confer some sort of benefit? So what we did was we had participants consume Kernza-based products for 2 weeks. We had a washout period in between, and then we had participants consume 2 weeks of whole wheat-based products. Our goal that we set out to recruit was 20 people. And then we also tracked GI symptoms over the course of this too, to make sure that there was no ill effects of consuming this stuff. One of the challenges that we're always faced with when it comes to studying diet microbiome interactions is that everyone's microbiome is very unique in terms of their microbiome signature. And so if you feed different people the same foods, they're going to have a different response in terms of what microbes change. And so it's really hard from a statistical standpoint to come up with any sort of consistent response in terms of a microbiome effect of any given food. So what we relied on for this study was what's referred to as the Gut Microbiome Wellness Index, which was this mathematical formula essentially developed by some researchers at Mayo Clinic. And the idea here is they essentially take all these different microbiome compositions from all this population data that we have and essentially turn it into this Gut Microbiome Wellness Index score. And this essentially allows us to reduce all this diversity down to this simple score. This is something that they've shown in vitro to be very responsive to different fiber types. And so this is also the first time that anyone has looked at this as an outcome in a diet intervention trial. So what we did, because we wanted to make sure to some degree that if there was going to be an effect, that we saw an effect, is that we screened people. People came in, they filled out a food frequency questionnaire, which essentially told us kind of what their typical fiber intake is. And so we selected specifically for the wrong thing. Very good figure. I need to update this. But so what we selected for was essentially a low fiber intake. So we wanted to make sure people who are already consuming a high amount of fiber probably had a large amount of diversity in the fiber sources. And so it was probably less likely that they'd experience a measurable change in these microbiome characteristics versus people who were consuming lower fiber. We originally set out to look for people who were consuming less than 10 grams of fiber per day. We had a hard time finding those specific people, a lot of people with like 11, and so we ultimately landed at less than 12 grams. But ultimately, so the way this worked is that people only did the intervention if they were found to be consuming a low fiber intake. What the intervention consisted of is 2 slices of bread and a half cup of puffed cereal per day. So it was either 2 slices of Kernza-based bread and a half cup of puffed Kernza or similar whole wheat products. So to find these people, we pre-screened and contacted for consent 146 people who expressed interest in the study. Typical with human studies, most of these people don't actually pan out. But we ended up consenting 51 people. There's always dropout at each one of these stages. We ultimately enrolled 44 people, which gave us 21 people that were eligible for the intervention, which aligned with our goal of 20 people. Study product compliance. So one of the things we want to make sure of is that people are actually eating this stuff. And so we found that this was really good. So oftentimes this was close to 100%. But overall, everyone consumed more than 85% of the study products. Baseline characteristics of our 2 groups, largely the same in terms of age. They were gendered oftentimes like a younger group. We're recruiting from a college campus. That kind of makes sense. In our intervention group, we actually had a pretty even split of males and females. Baseline calorie, protein, fat intake was similar. Differences, though, were observed towards the bottom here with fiber intake. We were particularly interested in fiber per 1,000 calories because this essentially controls for the amount of food that somebody is consuming. And so here we actually see this more meaningful difference in fiber intake.
The protein seems very low. 4 grams of protein.
Oh, that is an error. Yes, I'm guessing that is a decimal issue. That is a very, very low protein. So what we found, fortunately, there was no change in GI symptom scores with consumption of Kernza versus the whole wheat based products, which was good to see. And preliminary microbiome results, so we're still waiting on about half of the microbiome data to come back. But from the way that we will set this up is we'll do paired analysis to increase our power. That's kind of what we set out to do. That's why we often do the crossover interventions for microbiome stuff. But this is just what we have is kind of sporadic at this point. And so from the unpaired samples, though, the change in this gut microbiome wellness index is an encouraging trend in that we see an improvement in the microbiome wellness index score with the Kernza consumption that we don't see with the whole wheat consumption. Essentially with the whole wheat, it's pretty much, it doesn't really change versus with the Kernza, we see an improvement. This aligns with what we hypothesized in that people probably have whole wheat already in their diets. And so those specific fibers, whatever other phytonutrients are in there, are probably something that their microbiome is familiar with, versus the Kernza offers new fiber sources, new phytochemical sorts of things as well that might be impacting the microbiome in a positive way. Our paired analysis, too, again, similarly kind of shows this trend towards this improvement. We'll knock on wood that this holds. It's always hard with humans. You never really know what you're going to get. But overall, at this point, we expect the rest of the microbiome data to come back in the next probably like week or two. And so then we'll be able to do the full analysis, but we're hoping that this trend holds and that there is this very clear improvement that we see in this index score with the Kernza consumption. One other thing that we did was we also measured short chain fatty acids from the stool samples that we collected. What we found there essentially is that there was a lot of variability. There wasn't really a change in the short chain fatty acid production in either group. Short chain fatty acids are a metabolite that the gut microbiome makes, understood generally to be a beneficial metabolite. One of the challenges with measuring these in stool is that we don't actually know how much of these things have been produced and absorbed. And so it's not a great picture of what exactly is going on actually within the colon, but something that we kind of just tacked on as a potential metabolic endpoint as well. I think that's it in terms of the study update. Yeah, and at this point, at this point, again, knock on wood, trend seems to be positive towards there being an improvement in that microbiome wellness score with the Kernza consumption compared to whole wheat. But we have to wait for the full data set to come back. So I don't want to get out in front of our skis on that one.
So let's say hypothetically there is some sort of effect, something significant. How, if at all, could like a food company or someone formulating a product, how would they be able to talk about that? Because I know there's a lot of landscape around talking about nutrition, making nutrition claims. So I'm curious, how this could have an outside, how could be communicated?
So it would just be, and companies are very careful about the wording. You just can't make a health claim. And so you could be very intentional about the type of language you use, but kind of just alluding to like improvement in gut. I don't know exactly what you'd say, but essentially something along the lines of improvement in gut health or something to that degree that I'm sure you see the language on other products. It would be a similar sort of an idea there. Yeah.
What's your gut feel, pun intended? Like, what's causing this? Like, everything that you've seen, I feel like I always have a gut feel, like after I run an experiment, I'm like, okay, what could be driving this? Like, what's your gut feel?
Yeah, I mean, I think it probably comes down to, and then the question would be if people continued consuming this for like more than 2 weeks, if the effect persists. But I'm guessing what it comes down to is that people's microbiomes are familiar with whole wheat and they're not familiar with Kernza or as familiar with something like Kernza.
Yeah.
And so you're essentially just introducing new substrate that's available to the microbes, and so they can respond to that. And that's probably the shift that we're seeing. Yeah, and then the question being, is it something that would persist over time, or and what does it mean to this wellness index score?
Like past research on like like introducing new foods that change your gut microbiome, has that been proven in another context to be beneficial? Like
So we'll oftentimes see like there'll be a response if you introduce like a fiber supplement or different things. And so, but then the question though is like, what is this? So like the next step would be, is there like a physiological health benefit to this too? Is there some sort of like secondary benefit at this point? But I think as kind of a first pass, it's very encouraging that there does seem to be this distinct effect of, again, I don't want to speak too soon, but there does seem to be some sort of distinct effect of the Kernza-based products compared to whole wheat.
So the results you showed, were they after the week 2 or week 4?
So each one of the results are after each of the interventions, and then what they're being, it's not labeled exactly correctly, but essentially what they're being compared against is so there's 2 weeks before, and then there's a 2-week intervention. And so then we're seeing if there's a change from essentially the sample before the intervention to the end of the 2-week intervention.
So you said that this was incomplete yet, so we're not to week 6, we're not to the end of it.
No. So this isn't the complete set of samples. So the study is complete. So in each of these people completed the 2-week intervention, but what we'll do is we don't have all of the microbiome from people who completed both interventions, because we'll pair them together. So this is just the unpaired, what we have of
different people are coming in.
Yes.
At different times.
Yes.
Okay.
Yes.
And we had a question online from Jess, similar, I think. What do you think it is holistically that results in these improved benefits of Kernza?
Yeah, again, I'm guessing it's just fiber type, phytochemical content, that sort of the phytonutrient sorts of things. That again we could further interrogate at a later time point, but the overall phenomenon I think is encouraging at this point.
I think we might have talked about this when you gave your update last time, but did you balance the amount of fiber in the two, or is there
No,
more?
we balanced the amount consumed, but not the amount of. So the Kernza would have had more fiber just inherently.
Yeah, gotcha. How do you think reviewers will respond to that? Because there's kind of two things going on. There's a quantity difference and the balance and novel phytochemicals of things at the same time. So.
Yeah, and I guess what we're left with then is we're just unable to isolate what specifically, but the phenomenon of the one producing a difference compared to the other is still there. But yes, I mean that would absolutely be one of the limitations with interpretation is we can't interpret much more than that. But then we'd say the future directions would be trying to interrogate more specifically what component is driving this difference.
I think you probably explained it last time, but I'm not clear. For the gut, like health index.
Oh, yeah.
I don't really know. Is this a really big change, like a half a point?
No one really knows.
Oh.
But I would say it seems proportionally, yes. I would say like if we seem it's like a 0.25 in terms of the change. And so if we see like 1 is the kind of average or the typical, like that would represent a pretty substantial change from that value. But one of the challenges too is it's not super clear exactly like what sort of an amount of change you would be expecting, what's meaningful, because it's kind of a new metric as well.
Well, what's your background? You're a co-author on this paper.
Microbiome. The Dr. Sung at the end is the bioinformatics machine learning guy that developed the index, and then I just helped with interpretation of what it was doing.
So, but microbiome is like your core expertise, is that what you?
No,
Oh,
no, nutrition is core expertise. Microbiome is very much a kind of secondary.
So yeah, I mean, if you were to dream up, what's the follow-up study? You know, ample budget, what are the interesting questions? What would help make the link to the market? Was referring to, so any thoughts on where you'd like to see it go next, if you could?
Yeah, I mean, I think we talked about this in our original proposal too, but just the ability to use this as prelim data for a larger, I mean, it's so small, that it would be really interesting to see. One thing that would be nice to do essentially would be, both to assess a lot, if there's a longer term effect, if it's something that persists, but then two, to almost also see if, to kind of test in a way if the effect is real, is to kind of cycle people on and off of the Kernza stuff to see if it's something that's reproducible, if it's reappearing. Also, there would be the question of, if you control, could you isolate what component of it is potentially having this effect as well? But yeah, I mean, it's really valuable. It's really hard to do the intervention piece itself, and so like it's really valuable to have that as prelim data, and then yeah, I think there's any number of directions you could potentially go.
How hard is it to get like a fiber profile, the way you can get a fatty acid profile? Because part of the hypothesis is that it's a different profile than whole wheat. So is it difficult to quantify that, or straightforward to?
Yeah, I suppose I should probably be careful in claiming it's a different profile. I don't actually know that. I know the fiber content is higher. I don't actually know about the profile. But yeah, that I don't know. That's where George, and I think I saw Obed's name on there. That's where they would come into play and help with that aspect.
Yeah, cool. Any questions for Brian and Nick that were cut off when we transition?
Any plans for artificial cold screening? Or too early for that?
You know, we tried a little of that with the barley some years ago, Kevin, and I think that winter hardiness is a complex trait. You know, it's surviving ice sheeting, freezing, thawing, and I'm not sure the correlations have been so good with freeze chamber data and actual winter hardiness, but I'll have Kevin maybe comment.
I mean, the throughput is pretty low, you know, for breeding, I guess. I guess I've just gone with the idea of let's get it in as many different environments and do it outside and see if we can collect data that way. But when you get burned a couple of years, then you start thinking about alternatives. But Waleed is definitely, I've talked to Waleed about some things, and he's, in terms of like studying physiology and trying to identify, you know, like either some morphological traits or something like that could be screened for indirectly. I think using controlled environment things would be probably pretty useful.
I was also trying to think of like, could we do like a shuttle breeding to a similar place, like a high elevation Chile or something that might have a similar thing that we could bring it back on the timing, but I haven't gotten much further than that. But it would be interesting if there was some place in the world where we could get a comparable screen, and that the timing would be such that we could bring things back for a late September planting. But
in the barley program, it's been nice to see the results from all the different sites around the country, all that big collaborative network. Is there anything equivalent in the Durum world where you could just see how this performs in other geographies?
Well, let's see, for winter durum, there is a program down in Nebraska. And so we've done collaborations with Captain Prels. That's actually a nice area in need, because they pretty much don't get any snow that stays, and they pretty much can kill off most of the spring. Well, I'd say that for barley maybe, but maybe I don't know about durum, about whether they kill off all the spring durums every year, but it could be a good site as well. And there may be others too. We just have to maybe contact more winter cereals. I mean, Nick was at Colorado. Maybe there's some places in Colorado that are those holder that might be suitable. We'll have to look around for that. Or Germany with Friedrich Langen. I mean, maybe we can exchange more with him.
Yeah.
Oh, also there is a group. When I talked to Kurt Posniak, there is a group, I can't remember its name, in Saskatoon that is doing some winter durum in Canada.
Hmm.
So we tried to get seed exchange with them, but we just didn't have the time to get it through all the customs problems and things. But we'll try to, get some wider diversity of trial sites in the future when we have enough seed.
All right, well, thanks everybody for a great semester. Learned a lot, good ideas generated, and just fun to see everybody. And Anne's not here, but thanks again to Anne and Claire for the spread.